These are the back rooms of the Natural History Museum of Denmark. With 14 million specimens, the museum is housing one of the largest collections in the world, and there is a tremendous amount of unexplored data hidden in these collections, which we aim to preserve, extract and analyze.
We're interested in digitizing our collections because without online access to our specimen information, nobody can use the samples. And of course, our collections are here to be used. So we want to breathe life into them by digitizing them and making them available and accessible to researchers across the world.
My PhD project here at DTU Compute is thought as a start-off in making digital 3D models of specimens that were all collected at the Natural History Museum of Denmark.
What we're interested in is to look specifically at the biological and the functional differences between these polar bears in East and West Greenland and try and understand why they're different from each other.
So the overall idea for the project came from that we had a PhD student on his external stay at Berkeley University in California, and he saw how they measured skulls, and they use what is called a microscribe. So that's a small measuring device that you find landmarks on a skull, and it's a very manual process. And then he said, what about using the scanning techniques for this?
3D scanning is a focus area here at DTU Compute. We have two optical surface scanners, which both were built here. One of them is constructed for the purpose of traveling around, so the 3D scans can be done directly at the museum. In order to cover the whole surface, a skull has to be scanned in different positions. The final 3D model is then obtained by aligning and merging all these point clouds. Once we have digitized a large collection, as for example the polar bear collection at the museum, the overall goal is to describe the variation in shape of the skulls. And traditionally this analysis was done by having a look at manually placed anatomical landmarks. The resulting landmark coordinates can then be used to do some statistics. So for example, I could describe each of these two skulls, I could compare them to each other, or if I had a third skull, I could determine whether it looks more like this skull here or this skull here.
So the cool thing about this project is that we can obtain millions of points on each of the shapes. We can have millions of landmarks which we can use to do statistics, and then maybe we will find out some things about these shapes which we didn't know before.
It's almost the equivalent of how we, in the realm of DNA, have moved from single locus data to entire genomes. It's the same that before we had these single landmark data sets of the skulls, and now we can move to sort of have a spatial structure of the entire skull across many individuals, and I think that will really ramp up the insights and the inferences that we can make based on the morphology of species. If it works.
Yeah.
Of course, no. Yeah.